studied cells, and we feel that, and we know that the cells are a structural and functional unit of life. So why we are mentioning it? Because we have a diverse organisms starting from single cells to multicellular organisms. If you look into single cell organisms such as paramecium, euglena, and all these things, so the single cell is a life. That's why we are referring it as the referring cell as a life. I mean, structural unit of a life. And because that single cell performs each and every function of the life, including, say, the digestion, locomotion, respiration, assimilation, reproduction, everything is performed by a single cell. But at the same time, when we are talking about a multicellular organisms, so the work is divided into different cell types or different tissues. That's why when we look at our body as such, we are made up of many different kinds of cells, and different, they are having a different functions. So in order to do different functions, they have to have a different molecules in them. That's why each and every type of cell in our body is unique in nature. So when we are studying a cells, we use a tool microscope. So the microscope means we start from a very simple microscope to very high-end microscopes, from a bright field to confocal to, say, fluorescent microscope. Then we go for electron microscopes. So we use, hello? Okay. So we use a variety of microscopes to understand the cells. Basically, what actually we are looking in the microscope, what actually we are looking about a cell in the microscope. When we focus a cell under microscope, when we look at the cells under microscope, first thing what we do is we look at the shape of the cell. And the second thing, we look at the number of cells. What do you mean by shape of the cells? Cells are not of a same shape. They may be circular, they may be rectangle or a square, or they may be, say, spindle shaped or comma shaped. Cells means, please don't think that it is a cells of a plants and animals alone. It may be a bacterial cell, it may be fungal cell, it may be human cell, it may be animal cell, it may be plant cell. So every cell, if you look, they are of different shapes, sizes. Some of them are present in a small colony, some of them may be present in large number in a tissue. So, but shape we look. Based on the shape, we do identify many types of cells. Similarly, the second part, what we see is a number of cells. We count the cells. For example, if you go to the hospital, if you give our blood, they tell us what is the number of neutrophils, what is the number of basophils, what is the number of eosinophils or lymphocytes. Based on that number, so comparing with the reference number, so they will tell us whether we are having a normal range of number of these cells or not. So that is another thing. At the same time, we look at the size of a cell. Size of the cell, even if the shape is same, size of the cell varies. So we measure the cells using a technique called micrometry, and again we use a tool, the microscope. And we look at cell viability and cell death by maybe exclusion, dye exclusion techniques, or maybe using a fluorescent techniques. So we are able to identify whether cell is alive or cell is dead. And again, we may be identifying how the cell is dying, whether it is by apoptosis or whether it is by necrosis. And at the same time, using a different kinds of microscope, maybe higher end microscopes, we look at internal parts of the cells. Internal parts means it may be nucleus, it may be mitochondria, it may be Golgi complex, endoplasmic reticulum. So it may be exosomes, it may be some other vesicles which are present in the cells, or maybe food granules or color granules. So we look at various such internal parts of the cells when we are observing a cell under microscope. So basically, in addition to that, we look at we have already studied also meiosis, mitosis. In other words, we will be identifying what is the stages of a cell, whether they are in the interphase, whether it is in a division phase, whether cell is in prophase, metaphase, anaphase, telophase. All these things we are observing using microscope as a tool. In addition, if you look, so the expression of a product, say for example, if insulin is produced in a beta cells of Langerhans, how I can identify that insulin is produced in a Langerhans. I can have a specific antibody and I can stain it with the such antibody. Antibody may be tagged to the fluorescent color, or I can stain the antibody using an antibody, that specific molecule. And if I look at the expression, if I find some coloring changes, I can say that, okay, this molecule is present in this cell or not. So basically, we are identifying cells based on the properties. It may be a shape, it may be a size, it may be a number. So at the same time, we are looking at the cells in the microscope, whether they are alive or dead. So the internal part, cell cycle. So additionally, we do immunophenotyping also. What do you mean by immunophenotyping? For example, say if I take cells from the buccal cavity, if I smear and if I observe the cell under microscope, so based on the nature, maybe I can say irregular shape, the edges are actually tapering and the nucleus is present at the center. So based on that kind of a shape, I can say that this is a buccal cell, or this is neutrophil, this is lymphocyte. Like that, just by looking under microscope, I can identify a cell. Suppose if I take a RBC from my body and somebody's body, if I place it on the slide and if I try to observe, I may not be able to identify the blood cell type. So basically, in that case, we have to look for the markers which are present on the cells. For example, we are grouping the blood group as A group, B group, and AB or O group. So in other words, how we are dividing it? Because it is based on the presence of a specific molecules on the plasma membrane of the RBC. Suppose if RBC is having a antigen, when we are adding a antibody A, so antibody A will bind to the antigen A. Because of that, we will see agglutination, and agglutination we see as a observation. Okay, based on this reaction, if A antibody binds to A antigen of a RBC, we do see a agglutination. Similarly, antibody B binds to antigen B present in some of the RBCs, it gives a agglutination. If so, based on that antigen antibody reaction, we are able to identify the phenotype of a RBC or a phenotype of a blood, whether it is A group, B group, AB group, or O group. Basically, what I mean to say is that, so when you add an antibody binds to antigen. This is nothing but antigen antibody reaction. So what is our first inference? First inference should be the A antigen is present on the cells. So as the A antigen is present, our blood group is A. If both the antigens are present, then we say blood group is AB. If both the antigens are absent, then we say O group. If the antigens are, say, even the positive, negative, that is the B positive, A positive, all these things we will be identifying based on the antigen antibody reaction. So in other words, suppose if we are identifying a type of cell based on such antigen antibody reaction, we call such a phenomenon as as a immunophenotyping. So we do in immunophenotyping. In a nutshell, I want to say what are all these things: shape of the cell, size of the cell, number of cell, whether the internal parts present in the cell. For example, some of the cell may not be having, like RBC is not having a nucleus. Some of the cells may have a two nuclei. Some of the cells may have more mitochondria. For example, brain cells, heart cells, liver cells, muscle cells have a more mitochondria, lacks together mitochondria. Whereas the simple cells like lymphocyte may be having around 1,000 mitochondria. So the number is also very important. Not only the internal parts, as well as the number of cells also very important. Because say, for example, if you take a blood cells, if the number of blood cells is more, we may be inferring that it may be an infection, or it may be a cancerous, or there may be some abnormality. So based on the number, less number or more number, we can identify and we can tell the condition. So basically, all these things, whatever is listed in this slide, is nothing but properties of cells. So using microscope, so we are actually looking at the properties or characteristic features of a cell. So when we say we are studying a cell, we are generally we use a term cell biology or cytology. what actually we are studying in the cell. Study of a cells, we refer it as a cytology. Cytology is not just a study of cell, it is a study of properties of cells. What properties we study? Shape, size, number, viability or death, or internal parts, cell cycle, expression of a genes or the molecules in the cells, interaction of the cells, or you may be identifying a cells, or you may be looking for presence or absence of a specific molecules in the cells. All these things we look in a cells. And additionally, we may look for senescence, we may look for cytochemicals present in the cell by differential staining, or we may look for cell-cell interaction, maybe by cell junctions or maybe another thing, or we may looking into a differentiation aspects. Say, for example, cells may be de-differentiated into cancer cells. Suppose if you think of turning it back to the differentiation status, that is actually a cure of a cancer. Such kind of a things. Or maybe the after cell division, cell is differentiated into different lineages. So how they are differentiating, all these things we will be looking at, and all these are nothing but properties of cells that we are studying using a tool that is nothing but microscope. So we microscope, as I mentioned earlier also, starting from bright field to electron or photon microscope, we have a varieties of microscope. We use different microscopes for different purposes, but whatever it is, so we are studying the properties of cells. So cytology is nothing but study of properties of cells. Now I am using something else. Say, for example, I am giving my blood sample to the hospital where I am getting the number of cells and something, say my RBC is less, RBC is more. So if RBC is less, the people say you may be anemic. If the RBC is more, you are having a more RBC, or some cells may be more. So they interpret it in different way. But what is that actually they are doing in the hospital? They are actually measuring one of the property of a cell. What is that one property that they are measuring? The number of cells. So in this, whatever the example just now I have given is a number of cells. They are looking at the number of neutrophil, number of basophil, eosinophil, and all these things. This is true in research also. When we do some research, we do count the number of cells. So at the same time, even you can check the size of a cell. In many anemic condition, they look at the size of a cell. If the size of a cell is bigger, they say macrocyte. If the size of a cell is smaller, they say microcyte. Okay, so the size of the cell is also a parameter. And again, shape of the cell. For example, sickle cell anemia, if you talk, generally the RBC size is, shape is circular biconcave shape. In the sickle cell anemia, what is the shape of the RBC? RBC shape is changed. So in other words, shape is also a property of a cell. That is also looked into the in whenever you are measuring. So whatever you measure, whether you count the cell, whether you measure the size of a cell, whether you look at the measure the shape of the cell, whether you are measuring internal parts of the cell, the nucleus and all the things, or whether you are measuring the amount of molecule that is expressed in the cells. So that is, or you are identifying presence or absence. So basically, when you think of measuring any property of a cell, and you have to use a terminology that is cytometry. Cytometry is nothing but measurement of properties of cells. And again, what is the tool that we are using? We are using the microscope as a tool only. We are using the microscope to see the shape. We are using the microscope to measure the size of a cell. We are using a microscope to see the number of cells. We are using a microscope to see the depth and cell viability of a cell. We are using a microscope to observe the internal parts of the cells. We are using a microscope to look at other cells, other aspects of a cell, such as immunophenotyping, senescence, expression, everything. So anything that you measure in the cells is referred as cytometry. So cytology is study of cells. Cytometry is measurement of properties of cells. And what is the tool that we are using? We are using a microscope alone. Now, there are some limitations when we are talking about microscope. See, we cannot count large number of cells in a short time. So we cannot have a picture of those things. So there are many such limitations to overcome. So there are some advancements written as a flow cytometry. So what is cytometry? cytometry is a measurement of properties of cells. So when we are talking about flow cytometry is actually measuring a property of cells or any particles when they are flowing across the light in a fluid system. So you can define it as measuring a properties of cells or particles as they are flowing in a fluid suspension across an illuminated path is referred as flow cytometry. And the instrument used is called flow cytometer. So what we get from this instrument, there are three important things that we get from the instrument. One is relative size, relative granularity or internal complexity, and relative fluorescence. These are the three things that we get from any flow cytometer. So let me start explaining what it is later. So when we are looking at the flow cytometer has a three important components. One is fluidics, second term is optics, and the third is electronics. In the fluidics, we are actually suspending a cells. Generally, we have to make a single cell suspension, and single cell suspension means each and every cell should be single entities. If you look into our body is made up of organs. The organs are made up of tissues. Tissues are having many cells which are attached with each other. So that means you have to, suppose if you want to analyze a property of any tissue or anything, you have to take out and you have to separate the cells, and you have to have a single cell suspension for the analysis. So when you are having a single cell suspension, you have to suspend the cells in an isotonic solution so that cells will be retaining their shape, their structure, their everything will be retained if you are suspending a cells in a isotonic solution. Generally, people can use tissue culture media or maybe a PBS buffer or any such isotonic solutions to suspend the cells. And one important thing, they have to suspend in a single cell entities. So when you are suspending a cells in a fluid, then what actually happens in the instrument, the fluidic systems allows these cells to flow in a single line. That means like a queue. One cell moves after the next one. It's a queue. There is a queue in the cells. In other words, cells are moving in a queue in the instrument in a single line. When they are passing through the light, for example, so when the cells, for example, if you look into this system, look at this region. There are many cells here which are present together, suspended in a fluid. When they are moving in a system, they are allowed to move in a single line, one by one. So that is actually done by fluidic part of the machine. And the second thing, there are optical part present in the instruments. So what actually optical part? Generally, the optical parts includes lasers. Anyhow, I'll talk about that. So what actually optical part does is it actually shines the cells. In other words, it will illuminate it. Generally, the lasers are used as a device for source of light. So when the laser light is on in the machine, the light is passed through the cuvette there where the cells are moving in a line, so that each and every cell is exposed to the light. When each and every cell is exposed to this laser light, so the cells scatter the light and they fluoresce the light. This scattered light and fluorescent light is collected in the machine, and these collected light is converted into electronic signals, and these electronic signals, in other words the digits, are stored in the form of a values, and it is stored in FCS format in the machine. So now how the fluidic works? Say for example, if you look into this is a simple diagram on the right side. So where you can see, and at the same time, so you can see in the machine, there will be a place where there is an option for keeping a tube in which you have suspended as cells, and cells should be in a single entities, and you are having a single cell suspension. So when you switch on the machine starts aspirating, flow cytometer starts aspirating the cells from the tube. So as the cells are moving, so they started moving in a single line. So remember, they are moving in a single line. So there are some valves, okay? So these valves allows the cells to move in a single line. Say for example here, and the cells will move to the flow chamber. Once the cells are moved to the flow chamber, they are actually taken down and later on they are sent to the waste tank. So there are two chambers. One is flow buffer and another one is the cell suspension tube. So in the cell suspension tube, you are keeping the cells. Instrument aspirate this solution and allow this solution to move to the flow chamber. And at the same time, there is another chamber called the fluidic chamber. So here, so the sample is the fluid is nothing but the same fluid that you are using it for suspending a cells. This is also aspirated and sent to the flow chamber. So what actually happened here is that when the cells are moving in a capillary tube, so cells are haphazardly distributed, and when they are just injected into a flow chamber here, injected into a flow chamber, uh, they are moving at the center of the another fluid, which is nothing but a fluid that is coming from this tube. So that this fluid which is coming from nothing but a buffer which is surrounds the central sample, so that the central sample moves in a single line. In other words, this mechanism actually allows the cells to move in a single line. So basically this fluidic system is meant for making the cells are moving in a single line in this channel. As the cells are moving here, there is a point called interrogation point. So where the light is actually shines or illuminated the cells. So what is the fate of these cells? Remember, there are two kinds of flow cytometers. There are analyzers and sorters. I will talk about sorters later. So, but when we are talking about analyzers, see, the cells are moving like this straight away in the channel, and each and every cell is moving one after the other, and there later on the fate of the cell is we are sending them to the waste, and in the waste tank actually we are treating the cells with the sodium hypochlorite and we are killing it, we are destroying it. Now how this is a fluidic part actually. So in other words, it actually having a laminar flow and hydrodynamic focusing. Now, when we are talking about optics, so optics includes the excitation source and collection optics. See, excitation source is nothing but the laser source which produce a laser light. There are flow cytometers which have only one laser. Commonly, people use blue laser which produces 488 nanometer light. Some machines may have a 2 laser system. Some machine may have a 3 laser. Some machine may have 4 laser. And a higher end machines may have a 6 lasers or 8 lasers. Nowadays, we are having instruments which are having 8 lasers. So what is the use of this different lasers? See, these are monochromatic lights. So blue light means only the laser is producing 488 nanometer light. And if I am using, if the UV light is present in the machine means it is producing 375 nanometer light only. And if they are using a red laser, it is producing 633 nanometer light only. So that means if you are having only one laser, if it is producing a 488 nanometer light, then in the machine, in the cells, when you are staining the cells, you are supposed to use the fluorescent colors which are excited by blue light only. Suppose if the machine is having two laser, both 488, so blue light as well as a UV light, you can use a fluorescent dyes which can excite by both either UV or the blue light. Suppose if the machine is having a 5 lasers or 6 lasers, maybe nowadays we are having a violet laser, green laser, blue laser, the red lasers, UV laser, yellow laser. So we are having a different kinds of lasers. Suppose that means what actually happens is As the number of laser increases in the machine, so we can use multiple fluorescent colors for staining the cells. So that is the advantage of having many lasers in the models. So we are having a models which are one laser model, two laser model, three laser model, five laser, 10 laser. So that is the model. Then when the laser is produced, please look into this right side bottom line. See, this is a, for example, these are having a two laser system. One is blue laser and another one is red laser. So laser sources are this, they are producing a laser light. Blue laser is producing 488 nanometer light, whereas red laser is producing 633 nanometer light. And in the machine, there are collection optics. So these collection optics are actually the systems. They are actually made up of filters come mirrors. Okay, they do a dual purpose. They are acting as a mirrors, they are acting as a filters. And there are three kinds of filters generally they use in the machines. One is called long pass filters, and the second one short pass filters, and the band pass filters. In the machines, they may use a long pass and band pass, or short pass and band pass, or all the three may be used in a machines, and that depends on the model of a flow cytometers. So what actually long pass filters does, say for example, if this is say here in the picture right side, if you look into this, so dichroic mirror 560, this is referred as a short pass filter. That means what? Laser light 488 nanometer is coming like this, and it is going straight deviated and going here. And what is actually happening here? There is a dichroic mirror 560 short pass. That means below 560 is allowed to cross this mirror filter, whereas above 560 nanometer of light is reflected. So just now I said that dichroic mirrors does two functions. One is mirroring, another one is filtering. See, short pass filter means it allows the wavelength above the cutoff wavelength. Say, for example, below the cutoff wavelength. For example, if I say 560 short pass, that means below 560 is allowed to cross this mirror, whereas above 560 nanometer of light is reflected. Similarly, if I say a long pass, see, look at this dichroic mirror 640 long pass LP here. In this case, look at this, the light is coming here. So above 560 nanometer is allowed to cross this, and below 5640 is reflected. This is opposite of a long pass. Whereas a band pass system is band pass mirror, or the dichroic mirror is the one which cut off both left side and right side. In other words, suppose if it is a band pass, say 500, it will not allow, it will allow only the wavelength maybe from 480 to 520 nanometer to cross this. So below 480 and above 520 nanometers are reflected in the band pass mirror. So they use a different such kind of a mirrors to reflect the light and or the filter the light. So what actually happens in the machine is that, say for example, here the laser lights are produced. This is a flow cell. Just now I have shown here, this is a flow cell where the cells are moving in a single line like this. So you can look at the left side also here. And this is a place where this is called interrogation place, where the light is coming in a horizontal way. So light is coming in a horizontal way like this, you can see here. So this is the flow cell where the cells are moving in a straight line and the light is passing across the cells direction. As the light is coming here, each and every cell that crosses this interrogation point are exposed to the light. When they are exposed to the light, they do two things. One is scattering of light. What do you mean by scattering of light? Please look at the light, look at this image on the top. One is forward and the side. So there are, the first thing is the short angle scattering. That means if the, so you just imagine, take a ball put a torchlight from one side and observe the light from the other side. So it looks like a complete eclipse. Light is coming in from all the sides of a ball. This kind of a things happens here also. So depending on the size of a cell, depending on the shape of a cell, so you will going to see the scattering of the light when it focus on, when it illuminates or shines on the cell. This kind of a light, see for example, if this is a cell, this is a cell which is moving on the in a single line here. So when the light is passed on this cell, based on the shape and size, it produces a light or it scatters a light. That scattered light is actually taken forward, and this light which is taken forward is called forward scatter, which tells and generally the size and shape of the cell. Size may be different, shape may be same, or shape may be different, size may be same. So depending on that, we will get a different angle of light, this angle of light. And this angle of light will tell us, if the angle of light is very less, it means that the size of a cell is very less. If the angle of light is more, it shows that the cell size is bigger. So this kind of a light which is taken forward on the direction, say for example here the blue light is produced here, cells are moving in a same line, I mean in the straight line, and each and every cell is exposed to it. It produces, emits a scattered light, and this scattered light is taken forward only opposite to the laser line, and it is allowed to enter into one detector. This is called diode. Okay, so this kind of a scattering is called forward scatter. Forward scatter tells us the relative size of the cell. Now what is happening when the light is shining on the cell? So remember, there are many waves, there are many beam. It is a beam of light, and there are many rays of light coming in. Okay, so when such many rays not only touches the cell on the edges, but also because they are energized light, they also enter into the cell. When they are enter into the cell, the cytoplasm is made up of many particles. It may be a endosomes, it may be a Golgi complex, it may be a mitochondria, it may be nucleus. So these rays hit one particle, deviate little bit, hit another particle, deviate little bit, hit another particle, deviate little bit, or it may directly hit nucleus, it may deviate. In all the cases, the light is hitting the cell and it is deviating, and the light, depending on the complexity of the cell. Say for example, if the cell is very simple like a lymphocyte, lymphocyte has a big nucleus surrounded by a thin cytoplasm and the plasma membrane. When you shine the light on this lymphocyte, the light directly enters and hits the nucleus and goes straight in all the direction. That means right angle to the nucleus. When it hits, so it may go like this or like this or like this or like this, but it is a right angle to the hitting point. So in other words, it hits the nucleus and goes to the 90-degree or deviate to the 90-degree angle. Or suppose if it is hitting many particles in the cells, if the cell is very complex, hitting many particles, the angle of deviation will be more than 90-degree. So such kind of a light is taken here and it is diverted to another detector. PMT stands for photomultiplying tube, and it measures the side scatter. Side scatter is nothing but it is a scattering property. When the light enters into the cell, it hits many particles, deviates, and the light is coming out of the cell, and that light is collected and allowed to enter into another detector, and this detector is referred as a side scatter detector, or and this is giving us the complexity or granularity of the cells. So basically, when we talk about this system, it gives a scattering and the fluorescence. Scattering are of two types. One is forward scatter, another one is side scatter. Forward scatter tells the size of a cell, side scatter tells the complexity or granularity of the cell. And whereas at the same time, you may be staining the cells directly. For example, you are adding some fluorescent dye, for example, FITC, fluorescence isothiocyanate, which gives a green color. That means what? When the cells are stained with the green color, when the light is passed like this, when the cells are moving here, because already the cells are stained, the stain actually excites by 488 nanometer light and it emits the fluorescent light. The emission will be in the green color range. The cells are exposed to the blue color light, and these cells are, remember, stained with the FITC. The FITC absorbs this blue color and excited to green, excited and producing a green color light. And that green color light should be allowed to another detector. That means when the green color light of that particular cell is produced, the light is passing through. Remember, here there are filter. Okay, this filter is 560 short pass. Green color wavelength is 500 to 560 nanometer. So that means 500 to 560 nanometer of light is allowed to cross this filter, that is the dichroic mirror, and you are actually directing 500 to 560 nanometer of light to one direction, and this is going in direction, and you are keeping one more detector, and that detector is nothing but green color detector. So it detects the wavelength from 500 to 560 nanometer. In other words, it detects the green color produced by each cell. Suppose if you stain the cells with a red color, so what happens in this setup is, so red color wavelength is above 600 nanometer. So that means this is a short pass filter which actually reflect the red color light, and red color light is allowed to enter into another detector. So red color light produced by a single cell is going to another detector. Suppose if you are staining the cells with yellow color is allowed to enter into another detector, and orange color is allowed to enter into different color. Basically, the optical system allows the cells, whatever the light produced by a cells is sorted out. In other words, the wavelength light is the particular wavelength light rays are directed to particular detector. So there are, suppose if a cell, if you are staining the cells with a multiple color, the single cell may produce multiple light, and that means all the whatever the light that is produced, it will be detected here. If you are, and such kind of a diversion of a specific wavelength of light to specific detector is done by optics. So this looks very complex, but it is very simple. So looking into this system, look at this, there is a blue laser. When your cells are moving in a single line, these cells are exposed to the light. When they are exposed, they produce the, they show the scattering. Based on the forward scatter, you can detect the size of a cell, shape of the cell. Based on the complexity of the cells, you can detect that in a side scatter channel. And suppose if the cells are producing a fluorescent color, you can detect in different photomultiplying tube. So now what actually these photomultiplying tubes? These photomultiplying tubes, see for example here in the left side, the cells are moving in a single line, laser light is coming, and the cells when exposed produce a light, it is reflected and separate wavelengths are diverted. The blue color is going here, the green color is going here, yellow color is going here, like that red color is going to another side. So this is all made because of the dichroic mirror. So they allow the specific wavelength to go to the specific detectors. Based on the detectors, we can based on this, we can identify what is the fluorescence level are produced by a cells. Based on that, we can detect. So what actually electronical part does? So electronic parts is basically when the light is entering into the photomultiplying tube or a diode. What actually happens is the light is received by the PMT, and there is a ADC, analogous digital converter. So this PMT actually amplifies the light signal given by a cell. So if one, say for example, count 1 is entered into PMT. So in the PMT, there are many reflectors, many amplifiers. So 1 become 2, 2 become 4, 4 become 8. And like that, it amplifies, and finally the light signal is sent to ADC. ADC converts that light signal into electronic signal. So when the light signal is converted to electronic signal, equivalent, the electronic signal is nothing but a value. Suppose if the light intensity is very high, very bright light, equivalent value you will going to see as an electronic signal. If the light is very dim or very less intense, in that case the electronic signal will be very less. So if you look into the electronic part of the instrument, it gives a voltage first. That means what? Each photomultiplying tube, you can increase the signal, you can decrease the signal. Say for example, how it is operating. Please look into the left side down. Okay, so you can see this is actually a tube, the flow cell, where you are having a cells 1, 2, 3, 4. The first cell is almost hexagonal shape, a small cell. A second cell is a big circular cell having more granularity. Third cell is a small yellow color cells, okay, with the more granular condition. And the fourth cell is a big cell but no granularity. So what happens is when the cells are passed through this, so they produce a signal. So that based on the signal, say for example, as I mentioned in the previous picture, see there are photomultiplying tube. That is a detector for forward scatter, detector for side scatter, which gives a size and the complexity. Then the green color, yellow color, as well as orange color and the red color. Please look into this picture. So there are 4 cells. So you have a 4 detector, first detector, second detector, third, fourth, fifth detector. Say what happens is first detector detects the cells. It gives a fluorescent light, and the light is converted into electronic signal, and that electronic signal of a first cell is this much in a forward scatter. That means size of the cell is small. That's why the signal level is very less. In other words, intensity of light is less, the signal is less. Similarly, the third cell size is also less. So that's why the signal is up to this level. Second and the fourth cell, you look at the size and the size. They are bigger cells. The signal of the second and fourth cell is more in the forward scatter because it is detecting the size of a cell. Whereas complexity, in other words, the granularity, first cell is agranular. So the signal in the second channel, that is side scatter channel, is very less for a first cell and the fourth cell. Second and third cell are highly granular. Even though third cell is smaller in size, it is having more granularity. That means more complexity. Because of that, more particles are present inside the cell. Because of that, their signal is very high. It has given a more signal. You have stained the cells with the 3 colors. One is say red, yellow and green. One kind of a cell have taken a red color, another kind of a cell taken a say yellow color, third cells taken a green color. If you look into that, the green color cell produces a fluorescence light that is filtered and entering into the first detector. So first detector is nothing but fluorescence detector which detects the green color. So first and second, third cells are not producing a green color. So they're producing a very less intensity of light. Whereas the intensity of light is more in the fourth cell with respect to green color. Similarly, intensity of a yellow fluorescent light is more in the FL2 channel or the second detector, and the third detector red color is more, that is a second cell. So if you look into these pattern, you can make a plot in the computer. See, electronics includes the computer. So you are operating the machine through the computer, and you have a specific software, built-in softwares which are installed in the computer, and that is actually connecting the computer with the machine. And in the software, you can make such kind of a plots. So it is called dot plots or the scatter plots. So you can look at the right side, one more plot which I have mentioned. It's actually a WBC plot. So suppose if you look into the first plot, if I am having a forward scatter on x-axis and the side scatter on y-axis, you remember, look at the forward scatter and side scatter signal. First cell is smaller in size and agranular. So it is giving a signal very close to zero of x-axis and y-axis. Whereas second cell is size-wise bigger and granularity is more. So it is giving in the right-hand side corner, upper corner. So whereas the signal of the third cell, if you look, it is size-wise smaller. That's why x-axis is very less. Whereas it is highly granular. That's why it is y-axis is more. And the fourth one, it is giving a fourth one is actually a size-wise bigger, but granularity is less. That's why you will going to see the dot here. If you are having a plot of forward scatter, side scatter, where you want to look at the size and the complexity of a cell, you will be making this kind of a plot, and you are looking at where your cells are. And similarly, you can make any kind of a plot. You can make FL2 versus FL1, FL2 versus FL3, FL3 versus FL1, FL3 versus SSC, or FL3 versus forward scatter. That means you can look at the complexity versus fluorescence, fluorescence versus fluorescence. Suppose if you are staining the cells with a dual color and all. Say, for example, here in the right side, I have given an example of a blood cells. Look at this. You will find one group of cluster of dots here. Each dot is actually a signal given by one cell. Each dot represents a signal given by one cell. We refer this as an event. So if you look at this cluster of dots, they are very close to the x-axis. This is what I have taken on the x-axis is FSC, that is nothing but forward scatter, which tells us the relative size. Whereas y-axis I have taken as SSC, it tells us the relative granularity. So when I am looking at a forward scatter and side scatter, I am looking basically at what is the size, what is the internal granularity. Look at this. These cluster is telling me these are close to means 0, both on x-axis and y-axis. Theoretically, I know these are RBCs, maybe debris also. If I look at this population of cluster dots, this is actually the size-wise bigger than the first cluster, but it is not having any granules. If it is having a granules, y-axis, it should be somewhere here. But it is not having a granule, so on y-axis, it is very low. At the same time, on x-axis, it is little bit higher than the first cluster. So that means these cells are agranular cells, because remember, I am taking a WBCs. WBCs are having a lymphocytes, monocytes, macrocytes, eosinophil, basophil, neutrophils, and the agranular cells are basically the lymphocytes. So this cluster is basically lymphocytes. You can see the picture here also. And if you look at the little bit right side, you can see one more cluster. This is size-wise bigger than the lymphocytes. Similarly, complexity-wise little bit higher, little bit granular than the lymphocyte. These are monocytes and macrophages. And look at this big cluster. These are granulocytes, which comprises of majorly the neutrophils, then the eosinophil, and the less eosinophil and the basophils. Because if you look into the number of basophil and eosinophil, they are less in number when compared to the neutrophils. Neutrophils are hardly 60% of total WBC is actually the neutrophils. So this is how you will going to see the dots. When you are running a cell, you will going to see such things. Now, when we are looking at the fluorescence, what do you mean by fluorescence? Fluorescence means the cells absorb light, then they are excited, and they emit a light. In other words, they absorb a shorter wavelength of light, emit a higher wavelength of light. So that is what is detected in the machine. We are using a 488 nanometer of light to excite or shine the cells. If you are staining the cells with a fluorescent color, they are excited by this 488 and emitting a higher wavelength. That is, higher wavelength ranges. If you look into the WIPGR, we are using blue light. That means we are expecting green, yellow, orange, red, far red kind of thing. Some of the cells, remember, they are autofluorescent. You may be aware of a fluorescent cells. For example, GFP cells present in the green fluorescent cells, yellow fluorescent proteins, which are red fluorescent proteins present in some cells in some of the special organisms like jellyfishes and all, which are living in a deep sea, or cave dwelling animals, or some cells may produce the light. So they are having a cells which contain a specific proteins, and they are having a autofluorescence. So they are also excited by this light and they produce a autofluorescent light. So that is one kind. And another type of staining, what we do generally for staining a cells, whether it is any cell biology practical or anywhere, cell biology experiments, or maybe for flow cytometry, we directly stain. So far I was talking about direct staining of the cells with the specific dyes. Now at the same time we do use a fluorophores. For example, you just recall our earlier the where I have talked about identification of a blood group. What we do, we take anti-sera A, anti-sera B, anti-D, so which contains the antibodies A, antibody B, antibody for positive or negative of the blood group. So in other words, there we are not using any stain. What we are using, we are observing the agglutination as a observation. In some cases you may find precipitation, but routinely in all the cases you don't get such kind of a things. What you have to do is you have to have a fluorophores. Fluorophores are nothing but the dyes which are connected to fluorescent colors which are connected to specific antibodies. Say suppose if I take antibody A, if I add or if I connect the FITC stain or the fluorescent dye to that, when that fluorescent antibody A binds to antibody A, antigen A of the RBC, even if there is no agglutination, suppose if you observe under fluorescent microscope, those cells emit green color. So that means you are staining the cells indirectly. The cell surface, the plasma membrane may have many such proteins, and you may have a specific antibodies for that. If those antibodies are the specific molecules are attached to specific fluorescent colors, then when you add these antibodies to those cell mixture, these antibodies will bind to specific antibodies, and when these cells are passing through the light, they are excite. These antibodies which contain the fluorescent color excites and they give a specific color. So this kind of a staining also we do in the fluorescent flow cytometry. So either cells may be autofluorescent, or you may directly stain the cells or the molecules of a cell. For example, if I want to stain the DNA, I can use a DAPI, which will be excited by UV light, or I may use a propydium or 7-AAD, which will directly stain the DNA and which will be excited by the 488 nanometer blue light. So we can stain the cells in a different way: direct staining, indirect staining, or specific using a fluorophores, etc. And again, the selection of fluorescent color depends on the instrument. You have to see the instrument, what laser is present in the instrument. If the instrument has only blue laser, you have to select the fluorescent colors which should be excited by blue laser. If you are having many lasers, you can select any color which is excited by any one of these lasers. And when we are looking at applications, so analysis part, generally in the flow cytometry, we use dot plot, density plot, counter plot, histogram. But most of the time people use dot plot and the histogram. What actually dot plot tells you? Remember what I said is, I said each and every dot represents a property of one cell. And that property of one cell is, we refer that as an event. Say, look at this. This is one dot, means it is, this dot is produced by, or this signal is produced by one cell. See, in the instrument, what is the fate of a cell? The cell passes through the flow cell. It passes through the interrogation point. When the light is passing to, when the cells are exposed to the light, they emit they scatter the light, they emit the fluorescence. Then what is the fate of a cell? The cell goes to the waste tank. But the light emitted by the cell is entering into the detector. In the detector, light is converted into digits, and those digits are displayed here in the form of a dot depending on their values. If their values is close to the 0, you will find here. If the values are nearly a positive side, it may be on the y-axis or x-axis. So dot plot, you can take whatever the parameter that you take on x-axis, you will going to see the dot plot, the dots, depending on the parameter that you take x and y, you will going to see the dot plot. In the histogram, you will see only one parameter. X-axis is the only one parameter, whereas y-axis will tell you the intensity of a signal given by each and every cell. Say here, one cell is giving a less intensity when compared to the cell giving this very bright intensity here. So, but it is a intense that is actually displayed. And again, how many number of cells are giving that signal is looked at in y-axis. So these are the plot. Generally, dot plot and the histogram is used in the flow cytometer for analysis. If the dots are present in the low x-axis, low y-axis, we can consider that their expression is less on y and x. And if they are present in the high, similar to in the mathematical, like you can look at and you can see they are high in this. Similarly, see what are the applications? Why we need flow cytometer? See, if I ask anybody to count 100 cells in a microscope, definitely they count by taking maybe 10 minutes, 15 minutes. Suppose if I am asking them to count, say, 1 lakh cells or 10 lakh cells, it is very difficult for them to count a specific things. So for that purpose, in a flow cytometer, when you are running the cells, in about 2 minutes to 3 minutes, the machine can count 1 lakh to 10 lakh cells. So 1 lakh to 10 lakh cells properties can be saved within about 3 to 4 minutes in the machine. That is the advantage of using this machine. So what are the some applications? You can look at this, the right side. So you can look at the cytoplasmic granularity. Why cytoplasmic granularity is important? See, the cells may be agranular, cells may be granular. So you can identify that. Or someone may be doing a nanoparticle experiments. I'm giving one example. So they are taking one kind of a cells and they are running in the machine. So cells are showing a signal maybe lower. So you are taking a side scatter because you are looking at a granularity. And you are treating the cells with a, say, nanoparticle. You wanted to make sure whether the nanoparticle have entered into the cell. If the nanoparticles have entered into the cells, that means what actually happened to the cells? The granularity of the cells is increased. So if you are having a SSC plot on the x-axis, if the cell is moved towards positive side of SSC, that clearly indicates that the cell is granular. So that means cell from agranular state to move to a granular state, that clearly indicates that the granular, that means nanoparticles have entered into the cell. So in an experiment, you can clearly prove using this instrumentation that the cell is granular. So you can use this in the experiment. Then immunophenotyping. The best example I can give is, say, we know lymphocytes. When we are talking about the lymphocytes, there are many types of lymphocytes. Basically, we say B lymphocytes and T lymphocytes. Again, the T lymphocytes, if I talk, you have a cytotoxic cell side, then the memory cells, so helper cells, like different kinds of T cells are also there. So, but when you are smearing on the slide, when you are observing the lymphocyte, it is very difficult. It is impossible to identify which is B lymphocyte, which is T lymphocyte. How it is possible to identify them as a B lymphocyte and T lymphocyte? You can do it in microscope. You can add an antibody for T lymphocyte, tag to one color. You can add another antibody for B lymphocyte, tag to different color. And then afterwards, if you smear it on the cell, on the slide, and if you observe under fluorescent microscope, if one cell is giving a first color, second cell is giving a second color, you can say first cell is a B cell and the second cell is a T cell. So that kind of identification you can do both in microscope and here. But microscope has a limitation. You cannot do more than four types of cells. Very difficult to do because when you are using a fluorescent microscope, there is a leaching of the fluorescence and very difficult to identify many cells. But in this flow cytometer, you can add maybe 20 antibodies with 20 different colors. And you can identify presence or absence of those antibodies in the cells. That is the first thing. Second thing, as they are present, you can say that this cell is this type. Say, for example, T lymphocyte has a markers on the surface such as CD3, CD4, CD7, CD2, and all. And B cells have a CD19, CD22. And these antibodies are specific for this cell. Suppose if you add an antibody for CD19, antibody for CD3 with the two different color, and when you are passing the cells and when you are making a plot of two different color, and you can look at the positive and things, and you can identify the cell types. And this is true for identification of a cancer cell. This cell surface antigens or immunophenotyping is majorly used in a diagnostics also, other than regular research. People can look at apoptosis, necrosis, death phases. They can look at intracellular cytokine production, intracellular signaling, gene reporters, maybe using GFP, RFP, that is red fluorescent protein, yellow fluorescent protein, or any reporters you can see. Cell cycle analysis, DNA content and DNA synthesis, DNA damage and repair, senescence, cell proliferation, cell sorting. I will talk about this later. So you have so many applications. I have listed very few applications, and I will show you some of the examples also. So this is another thing. Please look at these graphs here. So the first graph, you can see forward scatter, side scatter. Same thing what I have explained earlier. See, this cluster is lymphocyte, macrophage, monocytes, and this is actually neutrophilic things. So when we are observing a plot in the machine, we can mark a specific cluster of events or cluster of dots. This marking is called gating, and the mark is called gate. So this is very important. Please look into this. So marking is called gating and the mark is called gate. You can see R1, R5, R6 is a gate. Okay. Now I have another plot here in the next side. Okay. Right side, where I have taken x-axis CD4, y-axis CD8. These are lymphocytes. Basically, when I am looking at a T lymphocytes, there are two types of T lymphocytes. One is helper cells, one is killer cells. Here I have given the example of analysis of helper cell and killer cells. So what I have done, CD4 cells I have are stained with CD4 is actually staining the helper cells. CD8 is generally present in the killer cells or cytotoxic cells. I have tagged CD4 with the FITC. It emits a green color, whereas CD8 antibody tagged with a red color. That means CD4 cells, that means helper cells are giving a green color. CD8 cells, that is killer cells, are giving a red color. So I have taken those detectors on X and Y axis. X axis green color detector channel, Y axis red color detector channel. Now what I have done, I have run the unstained sample. When I run the unstained sample, all the cells will be very close to zero. Based on that, I marked a line quadrant so that whatever the dots, if it is present in this lower left of both x-axis and y-axis, tells me negative side. Negative for the green color, negative for the red color. Whatever I am seeing here, this is upper left, which tells me these are positive for a red color but negative for the green color. This quadrant tells me these are positive for a green color but negative for red color. If I find a dots here, it tells me that they are positive for both green and red color. Now, if you look into this, these are producing only green color. Dots are present here. That means they are producing only green color. These dots are producing only red color. These are producing both. These are producing no color. So I can look at in the statistics of the instrument, upper left is this, is actually 789 dots are there. That means 789 cells are actually cytotoxic cells. And lower right is this, is about 732. 732 cells are actually thus helper cells. Whereas this one is lower left, 853 cells are negative for both. They may be natural killer cells, they may be something else. So basically now I know what is the number of this cell and this cell. This is extensively used for some kind of a diagnosis, such as one example is maybe HIV. So in the HIV, the CD4 cells are continuously destroyed by HIV, and the number of this cell is very less, and this can be used for diagnosis, prognosis of the disease. At the same time, in a regular experiments, so not only these blood cells, you can run any kind of a cell, and you can show what is the number of cells, positive or negative, and what is their percentage. Everything you will get in about 1 minute or in about 2-3 minutes when you are analyzing it. And I said you can use a histogram. These are all dot plots, but at the same time, you can use histogram also. Look at this. This is a dot plot representing the same data as this as that of the histogram present here. This is representing a cell cycle. These are showing a negative, that is apoptotic cell. These are cells in a G1, G0 state. These are S phase. These are G2 phase cells. So you will get to know what is the number of cells in each phase. So this data and this data is one and the same. But what I am using here is a histogram. You can use a, instead of the dot plot. It is up to you. You can use a histogram or dot plot. So important thing why I am showing is the gating. Gating will be done like this for the dot plot. Here gating is marking. Marking is nothing but a gating in this case. Now the application wise one by one, as I am showing here. See, when we are running a cells, we suppose if you are culturing a cells, you are treating a cell. Say, just understand that you are culturing a cancer cells. You may be working with anti-cancerous effects of a cell. So in that case, you can look at the effect of a drug, whether cells are arrested here or here or here. So you can look at where the cells are arrested. So you can make out what is the percentage of cells arrested in G1 or G2 or S. Based on that, you can detect whether the drug is effective or not. And at the same time, you can detect presence of a tumor in the sample. Suppose if someone is having a tumor, cancerous tumor, the tumors generally have a different amount of DNA. So you, based on the measuring a amount of DNA, you can identify a tumor versus normal. So that is called ploidy analysis. At the same time, if you look into the plants, plants have a polyploid condition. Some plants, generally people do breeding experiments where they cross the plants of different strains, and they will establish a plant. They wanted to know what is the amount of DNA present in that plant and what is the ploidy. So you can analyze by running a normal diploid plant and getting this peak, and running a test plant. Maybe you will be, based on the fluorescence, you can calculate what is the amount of DNA that is present in the plants. So cell cycle analysis will tell you the apoptotic cells which are having a less amount of DNA, and the cells arresting of cells at a different stages based on your treatment or interventions. It will tell you, you can identify a tumors, you can identify the ploidy condition of a cells. Similarly, you can look at apoptosis necrosis. One can use a annexin and propionimidate. Propionimidate stains the DNA. Annexin V stains the internal part of the plasma membrane. In apoptotic conditions, generally the plasma membrane flips out, exposing this serine moiety of that protein outside. Earlier, it will be present inside, but when it is turned outside, so it will be exposed to this. So you can identify apoptosis necrosis, cell viability. See, for example, these are apoptotic cells. Whether these are necrotic cells, these are living cells. Similarly, using a tunnel assay, you can estimate accurately amount of DNA damage. Accurately by doing a H2AX gamma staining, one can do double strand break repair assays, and you can identify double strand breaks in the cells. Similarly, in the research, one can do mitochondrial functions such as mitochondrial membrane potential, ROS, reactive oxygen species, mitochondrial mass, mitochondrial number, one can analyze. Similarly, one can look at a transfection analysis. Say, when you do the transfection, you may be using a reporter genes. So the reporter gene gives a fluorescence. It means when you transfect a specific gene to one cell or introduce a foreign gene to one cell, what happens is the efficiency is hardly 4 to 10%. That means only 4 to 10% of the cells of a total population is showing your expression of your required proteins. In that case, the reporter gene will tell you, you can use this for isolation and separation. The extravesicular analysis, something like a exosomes produced by a cells, that can be analyzed. So similarly, in the diagnosis or the blood cell markers, you can use a different kinds of a blood cell markers, and you can use it for a various disease conditions also. So I'm just focusing here the cells which are having a specific markers, the mature cells of our body, the blood cells I'm talking in this case. Similarly, each and every cell has a specific markers. So you can do immunophenotyping of it and you can identify the cell. Similarly, using such technology, one can identify different kinds of cancers, different kinds of leukemia. For example, if I'm talking about a blood cancer or the leukemia, there are more than 60 different types of blood cancers are there, leukemia are there. And some of them are treatable. And if they are treated well, if they are identified in early stage and if they are treated, the person will be healthy. So in this case, this system, the flow cytometer is a very good tool to identify such things. So these are some of the markers present in the cells, can be used for identification of the different cancers. So some examples I have given. Generally, people use it for the acute leukemia panels. They use some leukemia diagnostic markers such as T cell marker, that is T lymphocyte marker, B lymphocyte markers, or neutrophil marker, basophil marker, eosinophil marker, or natural killer markers. So they use a different markers. They will be having some panel of markers, and based on the positive and negative, they will identify the cancer type. For example, please look into this diagram. Here, they are actually getting a blast cells, the cells which are cancerous cells. So they wanted to see what is the proteins that are, what is the markers expressed in these cells. They are in this identifying presence of CD19 and CD10. So look at these, almost all these cells are positive for both CD19 and CD10. CD19 is a B cell marker. CD10 is a stem cell marker. In other words, cancerous marker. Similarly, CD34 is a hemocytic marker, hemopoietic stem cell marker. CD7 is again T cell marker. So that means these cells are positive for CD19, CD10, and CD34. Definitely, this is acute lymphoid leukemia case. So that is how they will identify. Similarly, I'm showing another case where CD13 is positive, CD33 is positive. CD13 and 33 is actually granulocytic markers. In other words, they are actually markers for either cancerous of macrocyte or macrophages or monocytes or a basophil or eosinophil or neutrophil. Again, you have to use some more markers. Say, for example, here we have used a CD14. CD14 is a marker for the monocyte and macrophages. That clearly indicates that this is a monocytic leukemia. And you can look at chronic lymphocytes also. Some cancers are chronic in nature. They are not acute. They are acute myeloid leukemia, acute lymphoid leukemia. Similarly, you can look at chronic myeloid markers also. And look at this. These are the markers. Using these markers, you can say very clearly the person is suffering from leukemia or not. And similarly, see, many a times there is a recurrence of cancer, and it is mainly because of the cancerous stem cells. So people wanted to use the cancer stem cells identification, because after treatment, person is normal for about 1 year, 2 year, 3 year, and suddenly there is a recurrence of the cancer. That is mainly because of the presence of the cancer stem cells in the body. That also can be identified using a specific dyes and specific markers using a flow cytometry. And many a times there may be a requirement. Say if the person is suffering from a bone, some cancers, there may be requirement of bone marrow transfusion. In that case, they take a bone marrow from the donor. They wanted to see how many stem cells are there, which hematopoietic stem cells are there, so that because they wanted to calculate how much hematopoietic stem cells they need to transplant to the patient. So in that case, you can identify and you can count how many CD34, that is a marker for the hematopoietic stem cell, and for the identification. and you can calculate and you can tell the doctor, and they will calculate what is the volume of bone marrow they need to be transplanted. That's why it is helpful in bone marrow transplantation also. And at the same time, please look at this. Everybody need to have this. Many a times we are not aware of these things. So our T cell count, normally we should have, say, CD4 and CD8 number. CD8 is generally less when compared to the CD4. So we need to, everybody will be, when we are checking CD4 and CD8, we will come to know what is the number of CD4 cells in our body, what is the CD8 number. Suppose if a person is having a HIV or any other condition, so there may be reduction of CD4 cells, and based on that, the doctor decides what drug need to be given for this kind of a patients, and they will regularly follow up this kind of a test. Neonatal sepsis is another thing which we can do regularly in the patient. See, when the baby is born, the baby may be suffering with some kind of a bacterial infections or something which leads to sepsis in its body. But detection of the bacteria in the body of the baby is very difficult because they have to isolate it. They have to take a blood, they have to culture the blood, and it takes about 3 to 4, maybe around 4 days or 5 days. In that case, if you take a blood, if you analyze, you can give the result in half an hour. So neonatal sepsis can also be identified. Ankylosing spondylitis is another condition where there is an accumulation of calcium in the backbone. So that leads to immoving. Actually, we are having a pain and we are not able to move, which can also be identified very easily. This is actually diagnostics things which you can do in the flow cytometry. Paroxysmal nocturnal hemoglobinuria, another condition where the person is urinating a red color urine early in the morning. First urine will be red because of burst of a RBCs. And that is a disease condition which can be identified using a flow cytometry. And different diseases can be identified other using a many such markers using a immunophenotyping in flow cytometry. So far, I was talking about analyzers, and there are some sorters. What do you mean by sorters? See, many a times, this is just an analysis. What actually happening in the machine is that cells are moving, they are exposed to the light, and they are emitting a light, and they are going to the detectors. And in the detectors, they are converting into digits, and you are analyzing the property. And what is the fate of a cell? Cells are going to the waste. But in our experiments, we may need some cells for our experiments. Say, if I take a blood, if I want to isolate only T lymphocyte, it is not possible for me using many techniques outside. So using this flow cytometry, I can separate the cells. I can have only T cells in my hand. So such kind of a flow cytometer which helps me to separate and which helps me to isolate a specific cells, and I can use it for culture or many other experiments. Such a flow cytometer is called FACS machine. FACS stands for fluorescence activated cell sorter. There are two kinds of FACS machines. One is mechanical sorter and electrical sorter. In the mechanical sorter, when the cells are moving in a single line, so when you do a gating, so suppose if you do a gating for a green cells, only green cells are isolated or pushed to one tube, and you can collect only one type of a cells. Rest of the cells are going to the waste. Such a sorter is called mechanical sorter, where only one type of a cells you can collect. Whereas in the electrical sorters, what actually happens is when the cells are moving, each cell will be forming one droplet, and each cell will be electrically charged. And they are passed through two different electrical plates. Based on the electrical charge, they are moving to different tubes. In other words, in electrical sorters, you can collect more than two types of cells at a time. So you can have those cells for your experiment, and which is not possible in a regular way. So such is a advantages and advance that we are finding in this. And nowadays we are having a flow cytometers called image flow cytometers. They are not only you are running the cells, you are analyzing using a plots, but you can take an image of each and every cell. So you can look at the image of the cells on one hand at the like a microscope, and the other hand you can see the sorting. This is the advancement that we are finding now. There is a mass cytometry which will again works similar to the mass spectrophotometer, which is now recently used extensively for analyzing the phytochemicals, molecules, metabolites, etc. And you can do such things using a mass flow cytometry. There is a full spectrum flow cytometry which works similar to the regular flow cytometry. Only difference is it gives every wavelength produced by a single cell. So this is another advancement that we see in a full spectrum flow cytometry. So I just stop at this point of time as the time is already more. Suppose if you have any questions, please ask. I have actually concentrated more towards the technical and analysis part. Thank you for patient listening. If you have any questions, please feel free to ask.
Thank you, sir. Thank you.
Thank you, sir.
Sir, can you please explain once more the difference between detecting a normal cell and a tumor cell?
Yeah, okay. So for example, can you look at this picture here?
Yes, sir.
Okay. See, normal cells generally express some mature cells have some marker. Generally, they express markers. Say, for example, CD45 have you seen this picture? Can you see this picture?
Yes,
Left
sir.
side, this right side, you can see cluster of dots.
Okay.
All these cells are brightly positive for CD45. Okay, that means all these cells have a CD45 on the cell surface. That's why they are giving more fluorescence. Whereas blast cells or the precursor cells, they are having very less CD45. And based on these differences, we can identify them as a stem cells. Okay. Suppose if you mark this, okay, I am actually marking this now. Now I wanted to know what are the other markers expressed in this CD45 dim population. Are you understanding my point? For that, what I did, in the same tube, I have added another antibodies actually. When I am running the machine, I mean sample, I have added many antibodies, not only CD45. I have added CD19 also. I have added CD10 also. Now what I do, I make another plot and I put a gate of this. I apply gate 1 here. Can you see here? When I apply gate 1 here, whatever the dots present within this region is displayed in this graph, second graph. And the second graph, what I am having is CD19 on x-axis, CD10 on y-axis. These cells, if some of them or all of them, if they are expressing CD19, they will show positive for CD19 here. If they are expressing CD10, they show positivity here. If they are positive for both, the dots will appear here. Based on that, you can say they are CD19 positive. These are CD19 plus CD10 positive. These are CD10 positive only. These are CD10 negative only. Hope this is clear.
So when you are applying gate, that means they are only selecting the blast cells, right? They
Right.
are
Here in this case, we are selecting a blast cells to identify a cancers. Okay. This is true for everywhere. Say, for example, that is what I was talking about gating. Here I said, right? What I did, I marked this region. These are lymphocytes. Can you see this? These
Yes,
are
sir.
lymphocytes. And I applied this gate to here. So I wanted to say how many are CD4 and how many are CD8 out of this lymphocytes. In other words, T helper cells and T cytotoxic cells. So when you apply gate to this graph, that means you are looking for events or the dots or the cells signals which are positive or were present in this region. That is the main reason of a gating. So based on that, you can identify different kinds of cancers. That is one aspect. So you can use a different kinds of a marker. Say, for example, here I have shown a different marker, developmental marker. Say, for example, if you want to say if the cells are positive for cancer cells, suppose if you are identifying, if the cells are CD19 positive, then you look for CD5. If they are positive for CD5, then you look for another markers, some more markers. If these CD23 is positive, then there may be chances that the person is suffering from chronic lymphocytic leukemia or small lymphocytic lymphoma. If the CD20 is positive, whereas CD23 is negative, then the person is suffering from either mantle cell lymphoma, lymphomatous polyposis. If the CD5 is negative, then again you have to go for CD10 positive, CD10 negative, CD11 positive. Again, CD10 positive means you can think of follicular lymphoma, Burkitt's lymphoma, hairy cell leukemia. Based on the markers, you can identify the different kinds of cancers. I am talking here with respect to blood cancer, but it is true with all kinds of cancer. The people have worked with the brain cancers, liver cancers. All these markers are available and they are identifying using such kind of a thing. I have given just as an example to show how one can utilize it. So basically, you can use this for diagnostic purpose, HIV monitoring, or cancer cell identification, or cancers identification, or tumor identification, or tumor cell identifications, or you can use it for hematopoietic stem cell analysis and may be helpful for transfusion, or you can use it for neonatal sepsis condition, or you can use it for ankylosing spondylitis, paroxysmal. All these are diagnostics. These are all diagnostic aspects. But in research, definitely you can use this for sorting, because many a times we need many stem. For example, if I am working with the stem cells, I can take organ from any animal or maybe the human or human tissues, or I can take. But out of 10 lakh cells, I may get hardly 10 to 20 or 100 stem cells. Suppose if I want to isolate only those stem cells out of 10 lakh cells, it is very difficult without this mission. What I do, I use a specific marker for stem cells. I'll make a single cell suspension. I use a specific marker. These markers will bind only stem cells, and thus it is a rare events again. So I am running the all 10 lakh cells in the machine. I am taking the 100 cells are positive for the markers, and I can separate out and I can use that cells because they are stem cells, and I can use it for my experiments. So such kind of things you can do using this machine. Even stem cell therapy and all, the people do like this. They will isolate the stem cells, then they multiply and they make maybe a layers or something. They may use it for therapeutic purposes, even diagnostics as well as therapeutic purposes they use, as well as research purpose also. These are some advancements present of the recent advancements in the flow cytometry.
Thank you, sir.